Good day, students. Welcome to the last lecture recording on Module 6, Energetic Reactions and Food Enzymes. In this section, we're going to focus on the use of enzymes in the food industry, where we talk about the sources of these enzymes, we talk about the specific enzymes and what functions do they perform in processes and products, and then the advantages thereof of these enzymes compared to your traditional processing methods. Okay, so just to recap what we said about enzymes in the previous recordings was enzymes are biological catalysts that then speed up chemical reactions. And then due to this ability to catalyze reaction, enzymes are found being used in different industries like the leather industry, the pharmaceuticals and detergents industry. However, as food technologists, we only focus on the application of these enzymes in food, and we find that these enzymes find widespread application across the different food industries. Enzymes are used in the brewing, in baking, in the dairy industry, in processing of meat and fish products, as well as in beverages like your teas and then your fruit juices. And lastly, they are also applied in agriculture. So when they are applied in these different food industries, they modify and improve the existing functional, nutritional, and sensorial properties of those food products. And the reason why enzymes are actually chosen as an alternative to chemical based processing would be due to the fact that enzymes, like we already mentioned, they are specific and they produce specific products from a certain substrate. So therefore, due to that, there are then few side reactions and waste products forming as a result of using enzymes. And we had mentioned that enzymes lower the activation energy required to kickstart a chemical reaction. So therefore, instead of using high temperatures and chemicals to catalyze a certain reaction, then it means that by using enzymes, you lower the energy consumption, you lower the emission of gases, and then also, since you're also not using chemicals that would usually be found in the drainage system that results into going into streams of oceans and seas, whereby it's going to contribute to pollution. When using enzyme, there's no such thing as pollution, because we had also mentioned that the enzyme get used again. So it's not as if it can be thrown away. It can be used for another substrate that has to be catalyzed. So in that way, that is why we then prefer using enzymes. We can usually say it's a green processing technique compared to the chemical-based processing techniques. So when enzymes are actually used in food products, they come in two major forms. We have what we call endogenous and exogenous enzymes. So endogenous enzymes, these are enzymes that are functioning within a microorganism. So like microorganisms like your yeast, your Lactobacillus lactis for yogurt production. So when those microorganisms are fermenting those components to produce this certain type of products, it's actually the enzymes within the microorganisms that are doing the function. So that's why we call that endogenous enzymes. But when we talk about exogenous enzymes, we talk about enzymes that have been extracted from that source or produced from that microorganism, extracted, and then will be added in a food product or food processing. So of those commercial sources of enzymes, the enzymes are actually obtained from either your plant, animals, or microbes. Okay, so those industrial enzymes that are actually obtained from those three sources, 50% actually comes from fungi, 30% from bacteria, 12% from animals, and 8% from plants. So based on those figures, we actually see that or conclude that the microbial enzymes are the mostly used in terms of food production. And the reason for that is that in terms of producing enzymes from microorganisms, it's a cheap process. All you need is just your agar and your nutrients, favorable conditions that are obviously produced by incubation, and then voila, you have your microorganisms growing and producing those enzymes. Meanwhile, with the plant, you have to plant it, wait for it to grow, and then you can harvest that. But also the growing of that plant or the animal for that matter will then be dictated by the environment. Did it rain? Was there enough water? Was there enough sunlight for photosynthesis? Was there enough plants for that animal to graze in order for the animal to grow? So in that way, it's not cheap in terms of the plants and the animals, but it's cheaper for your microbial enzymes. And the fact that they are produced using controlled environmental conditions, the enzymes that is formed, or the products, are then predictable and you can control them. So each and every time when you're producing a batch, it has constant composition. Meanwhile, of the plant and the animal will be affected by what the plant is, or the animal is eating, or whatever you are feeding those two components. Like is there enough sunlight, like I already mentioned? Were the plants watered, and so on. And lastly, one of the most important characteristics of those microbial enzymes is that they are heat stable. So meaning that they can be used in processes whereby high temperatures are required. An example will be your thermostable alpha amylases that are used during production of your glucose syrup from starch, whereby temperatures as high as 80 degrees Celsius are used, and at those temperatures that thermostable alpha amylase does not denature. Meanwhile, other enzymes will denature easily at 60, 50, 40 degrees Celsius. And then lastly, they work at a broader pH spectrum. So unlike certain enzymes like your pepsin, your trypsin, those ones would work in an acidic environment only, but your microbial enzymes can then work in acidic up to slightly alkaline conditions. Therefore, you can actually use them in a range of different products depending on the pH. So as we had already mentioned that microbial enzymes constitute 80% of the total production, some of the specific microorganisms that I use in terms of extracting these enzymes from are your aspergillus, bacillus, and other Saccharomyces. Those are usually your yeasts. Okay, so then in the next two slides, that's where we talk about the different, because we have already mentioned the sources, the different sources where we get these enzymes. So in these two tables, we have a table tells you about the source, the source type. It will tell you about the specific name of the enzyme and where in that source is that enzyme obtained, and then the specific product application. So for the next two slides, you need to remember and know those sources as well as the product application, but most of those we'll talk about that as we go in the next slide. So from here onwards, what we dwell on specifically are the different types of food processing industries, and then from there we'll talk about the different enzymes that are used in that industry and name them and their specific functions. So the first industry will be the baking industry. So we already know when it comes to baking, when you bake, you need your flour, you need your water, you need your salts, and sometimes you also add your sugars and also your yeast and stuff like that. But now breaking it down further, you know that your flour, which then contains your protein and your starches and so on. So what happens is that in baking, because baking involves both your carbohydrates, your lipids, and also your proteins, and then they form a network. Not each and every of these components are acting on their own, but they all act together, and products from one molecule affects the process or the following process in another macromolecule within the whole food system or the baking system. Okay, so the first enzyme that we're going to talk about is the enzyme known as glucose oxidase. Okay, so glucose oxidase, what happens during baking, the whole entire baking processing from mixing up until the actual baking of the actual product, glucose oxidase act as what we call an oxidizing reducing agent. Okay, so let's just recap a little bit when we spoke about the gluten protein network structure, whereby we mentioned that gluten is made of glutenin and gliadin that then form together, especially during kneading of the dough. and then cross-linking of those due to their cysteine double bonds then obviously results in the gluten structure. And then we mentioned that we could actually modify the structure of the dough or the functional properties of the dough by either adding your reducing and oxidizing agents, which are chemicals like your potassium bromate and then your ascorbic acid. Do you still remember that? So now, instead of using those chemicals, bromate and ascorbic acid, vitamin C, now those two are then substituted by glucose oxidase. So what does glucose oxidase do in terms of the baking structure or the baking network? So what glucose oxidase does in the presence of glucose and oxygen, then it converts your glucose into gluconic acid and then hydrogen peroxide as a byproduct. So what the hydrogen peroxide does to the gluten structure, because we all know that hydrogen peroxide is an oxidizing agent. So hydrogen peroxide oxidizes the cysteine bonds and then it results in the cross-linking of the cysteine bonds. And by doing that, it's actually strengthening the dough structure and it's changing the dough structure. So by changing the dough structure, as we already mentioned, it actually changes the functionality. So we said what it does is it cross-links because the oxidation obviously induces the formation of the sulfur disulfide bridge from your reduced form, and it becomes oxidized, and by oxidizing it, it strengthens it. It actually improves the structural properties that are desirable in bread making because it helps in terms of the volume, the loaf volume during baking when the proteins coagulate later on. Okay, so usually this is beneficial usually when you have flours that contains weak protein or low protein. So by cross-linking the protein, they strengthen the protein. So that will be that. So that will then affect how the bread volume will increase during baking. Okay. So other enzymes that are also responsible will be your proteases. Your proteases, what they do is, instead of working on the sulfhydryl group, they don't do that because proteases work on the actual protein by breaking down the peptide bond. So these proteases, what they will do is that they will reduce the chains, especially during fermentation, during your retorting stages. And then by doing that, they break the links between some of the amino acids, and then what it results in a gluten structure that is much softer, then that can also be applicable for different type of food products. And as a result, it reduces the mixing time of the dough because now the gluten structure is also being interrupted. So all these enzymes are applied in terms of when you are looking for a specific type of product. So when you're looking for a more flexible dough, then you can use your proteases. If you want a harder dough structure, then you can use your glucose oxidase. And another one that is used in the baking, especially it's used is lipoxygenase, that helps in terms of bleaching the natural pigments that might be found on the flours. Okay, so as we had already mentioned, that baking is not just the protein on the other side and the carbohydrates standing there in isolation. Everything comes together and forms the whole network. Okay, so other enzymes that are also involved in baking would be, in terms of your baking, we already know that the main component will be the flour, and the flour, as we mentioned, is made up of protein, starches, and other smaller components and sugars. So as we already know, starch contains obviously your amylose, your linear amylose structures of glucose joined together, and then also your branch amylopectins. So during baking, what can happen is that your alpha amylase are actually introduced into the baking network, and for the reason for that is because, remember, your alpha amylase are going to break down the starch. So when it breaks down the starch, it breaks down the starch, and then by breaking down the starch, these alpha amylases form your dextrins, such as your maltose and maltotriose and some limit dextrins. Okay, and then further down, because these then should also be further broken down again into simple sugars that are obviously going to be required by the yeast. So then the second type of enzymes that are then used are what we call the glucoamylases, that will then break down those maltose, maltotriose, and those limit dextrins into simple glucose sugars. So when those glucose sugars are now available, then the yeast can then use those glucose sugars and produce carbon dioxide. And that carbon dioxide is the one that's actually responsible for the low volume, because as the gases is produced, then we have the gluten structure that is now soft and extensible due to either oxidation or reduction. And then what happens is that the bread rises because of the what? Of the extensible dough. And then that's when during baking, when that has risen, then the bread then coagulates and form that bread structure. So also the other important thing about those glucoamylases is that they obviously produce fermentable sugars, and these fermentable sugars are your simple glucoses. When that glucose is formed as a simple sugar, then it reacts with some of the protein amino groups, and then it forms the Maillard reaction, and that brings about the browning, the crumb structure, and then the flavor, the smell of the bread comes from those protein reacting with the sugars. Okay, and some of the other enzymes that can also be used in baking will be asparaginase. Asparaginase is an enzyme that breaks down asparagine. Asparagine is an amino acid, and we had mentioned that the problem with asparagine in the presence of protein is that it can result in the formation of acrylamide. Remember, we mentioned acrylamide forms via the Maillard reaction, but it can cause cancers. So by actually degrading the asparagine using asparaginase, you actually prevent acrylamide formation or the Maillard reaction via acrylamide, but then you still form the brown flavor via pure Maillard reaction. So you can already see that the properties that have been changed due to dough structure are then also affected by the fermentation, whereby when the yeast ferments the sugars, then carbon dioxide is formed, which then aerates the dough. And if the dough is hard and cross-linked, it actually reduces the volume. But if the dough is extensible, and extensible, it means that the low volume can actually increase. So those will be the functions of those enzymes within the baking industry or the baking complex. Okay, and then second, we come to dairy. In the dairy industry, so enzymes are used for different products. I mean, in the dairy industry, we have cheeses, we have milk, the amasses, cultured milks, we have different types of cheeses, and then we also have the milk itself, and then we have ice cream. So many products that results from incorporating dairy or producing different types of products. So the first product that we'll talk about will definitely be cheese, your simple cheddar or Gouda or Gouda cheese, whatever you want to call it. So those are actually made by coagulating your protein, your casein protein, and then separating the curd from the whey protein. So the enzymes that are used there is known as Rene. Rene is an enzyme mixture that is actually extracted from the stomach of calves, baby cows. So it's extracted from those, and then Rene is actually a mixture of enzymes. The main major enzyme in there is chymosin, which is renin, which helps in terms of the coagulation. And then renin in the stomach of your calves helps the calves to be able to digest the milk protein. Okay, so then what happens is that that Rene coagulates the casein and then forms the curd that can be pressed and brings about the functional properties of cheese. That is why your cheddar or your Gouda cheese is now firm solid because of those curds, but it comes from milk. So it means that the milk functional properties moved from being liquid to becoming solid, thereby producing a product that is desirable for consumption. Okay, and then the second type of enzyme, so your chymosin are usually what we call exogenous enzymes because they've been extracted and then they are added. And then also something else in terms of cheese ripening. So the cheese has to ripen by being stored at certain conditions for a certain period of time. So what happens is that the enzyme lipases can be added there, exogenous enzymes, or those lipases can be produced by the microorganisms, and that is found usually in those cheeses like your Roquefort or Camembert, whereby those molds produce those enzymes that will then break down the fat. So the lipases are beneficial during ripening of cheese, and it brings about the flavor because of those free fatty acids that are liberated from the triglycerides. And then second types of enzymes will be your proteases that are also endogenous. Those are also involved in the aging of cheese, and they bring about the flavor production because of those large proteins are then broken down into small peptides, and they bring about the flavor of cheese. So that is why the different cheeses have different flavors based on the type of enzymes that are produced there. Okay, and then we come to another set of enzymes, the lactase. Lactase is an enzyme that breaks down lactose. So each and every human being, or most human beings, have lactase that breaks down the sugar, milk sugar lactose into glucose and galactose. But some people lack that enzyme. So that is why some people say they are lactose intolerant. So in that way, they usually substitute their cow's milk with other soy milk or almond milk because they cannot digest the lactose. But thanks to lactase, the lactase can be added in milk, whereby it catalyzes the hydrolysis of your lactose into galactose and glucose, and in that way people that are lactose intolerant then now can drink that sugar. And usually lactose free milk is much more sweeter because of the liberation of the glucose from the galactose, from the parent lactose. Okay, another important thing is that the lactose, when usually in milk powders, it actually results in the caking due to crystallization of the lactose. But by using lactase to break down the lactose, then the crystallization is reduced. So that is also an important aspect, especially during storage, where crystallization and caking can take place. Okay, another set of enzymes that are found in milk, that are inherent in milk, you have lysozyme. The lysozyme reduces bacterial count. We also spoke about lysozyme being found in eggs, remember? So what happens is that lysozyme is specific, that it breaks down the peptidoglycan layer. So those remember, the peptidoglycan layer is actually sugars that are cross-linked by proteins. So what happens is that that wall that makes up the peptidoglycan can actually be lysed by the lysozyme, and in that way it becomes an inherent antimicrobial or preservative, we put it that way. Okay, and then also what happens is that another type of enzyme that can also be applied in the dairy industry is trypsin. Remember when we spoke about trypsin inhibitors? Okay, we call them anti-nutritional factors. So trypsin is obviously found in the stomach, and then it is a protease that breaks down proteins. So what happens is that trypsin can actually be used to pre-digest casein in baby milk formula, because babies cannot digest casein yet. Okay, so because they don't have those type of enzymes, strong enzymes to do that. So as a result, then if trypsin is used to pre-digest the casein, then when the babies drink the baby formula, when it's made up, the casein is already broken down. So in that way, they can easily further digest that. So it's actually useful for that. And then the last set of enzyme is catalase. So what catalase does is catalase degrades hydrogen peroxide to form water and oxygen. Where it is very important is during production of UHT milk. sterilized milk, what happens is that the Tetra Pak that is usually used for packaging of those milk is usually sterilized using hydrogen peroxide boiling at high temperatures. So that one becomes sporocidal. It kills all microorganisms. However, after those Tetra Pak have been flushed with that hydrogen peroxide, then what happens? The hydrogen peroxide, obviously traces will be found in milk, but then catalase is then used to break down that hydrogen peroxide to form water and oxygen, and in that way it does not become harmful to the consumer, or traces of it is not found. And then we come to the meat and fish industry, or producing of meat and fish products, whereby we rely a lot on proteolytic enzymes in terms of their tenderizing role. When you tenderize the meat. So we usually call this one sulfhydryl proteases, because their active sites are actually made up of your cysteine and histidine residues, which obviously are your sulfur containing amino acids. So of the main tenderizing proteases that are applied in the food industry, we have what we call ficin, bromelain, and papain. So ficin is actually they're actually all extracted from plant sources. So ficin comes from figs, bromelain comes from pineapple, and papain comes from the leaves of the papaya. So what they do as proteolytic enzymes, they break down the adhesions of proteins, and they are very beneficial during aging of meat. I remember when we spoke about aging of meat, that after rigor mortis, or after slaughtering the animal, rigor mortis sets in, and what happens is that the actin myosin complex then forms, and it becomes hard, and that way that the meat loses its water binding capacity. So the meat then has to be aged at certain conditions for a certain time. Usually those conditions are different temperatures, and in that way that adhesion is lost. But what we can actually do is these enzymes are then administered to a live animal, via the veins, and injected into the animal before the animal is slaughtered. Okay, and then in that way it can tenderize the meat. Then you don't need to even age the meat. You can actually use the meat immediately, because now those adhesions will be then broken down, and then the meat won't be as stiff as it would after any slaughtering. Okay, so what these enzymes are actually used for, besides meat tenderization, in terms of some of the marinades that are used out there, they are also used in terms for removal of meat scraps from bones. Usually in the meat industry, nothing gets thrown away. Remember, we said the bones are going to be used to extract your collagen. However, those meat scraps that are actually adhering there are what they are used for products like your polonies or your Viennas and stuff like that. So in order to obtain maximum of those meat scraps, so those proteases are used, therefore then they separate those meat from the cartilage and the tendons, and thereby increasing the yield. And then some of them, and not only plant proteases are used in terms of the meat and fish industry, they also use microbial proteases like your Bacillus subtilis and Aspergillus oryzae. So the Aspergillus oryzae, remember oryzae comes from maize, so those are enzymes, those proteases are then extracted from those two microorganisms. And then we also have some other enzymes like the chitinase, cellulase, the proteases that are actually used in the fish industry, whereby they are used to actually loosen the shrimp shells, like shrimp, not the one that you find with the shells when you go eat at Ocean Basket, but those shrimps that are actually ready to cook or ready to fry or whatsoever. So if that has to be done manually, it means that it's going to take a lot of time, and then obviously a lot of hands. So obviously the production will be quite high. But by using the enzymes such as cellulase and chitinase, what happens is that those shrimp cells can be loosened, and then that will be the separation of those cells from the actual shrimp. Isn't that cool? Okay, and then we come to the confectionery industry. The confectionery industry, whereby they produce syrups, and those syrups are used also in baking, and they are used in sweets and whatsoever. So here That's where we talk about starch, whereby your corn starch or potato starch or any type of starch are then hydrolyzed to form syrup. Okay, so your amylases or glucosidases are very important there, because during liquefaction and saccharification, what happens is that the corn starch is hydrolyzed to form the corn syrup. Okay, so in the process, when alpha amylase is actually used to hydrolyze the starch, it produces some 10 to 20% glucose. But when glucoamylase is then used, it produces 93 to 96% glucose, which is then used in a range of products in the food industry. Okay, and then the most important one was the obtaining of the new enzyme glucose isomerase, whereby you already know that it isomerizes glucose and then forms fructose, and by that then we result in what we call high fructose corn syrup, and not high glucose corn syrup, high fructose corn syrup. And very important about the glucoisomerase, they are highly heat stable because of obviously the high temperatures that are applied in these processes, and they are not affected by the substrate concentration. Remember, we mentioned that when the substrate concentration increases, the enzymes become saturated. Remember that? But with glucose isomerase, no matter how much the substrate concentration, it does not affect how the enzyme function in catalyzing the reaction. Okay, so of the glucose isomerase, we have the invertase, which then converts sucrose to glucose and fructose. And then the one most important aspect of this invertase is that it prevents sucrose crystallization, because sucrose crystallizes very easily, but when it's cleaved to glucose and fructose, then it does not crystallize as it does with sucrose. And then we also have in the fruit juice industry or beverage industry, where we have the importance of using pectinase. So pectinase is an enzyme that works on pectin. So what does pectin then do? Pectin, when juices are extracted using different types of extraction machines in the juice extraction industry, the pectin then causes clouding in those beverages, especially your apple juice. The apple juice looks all cloudy, and that also, the releasing of pectins and other insoluble carbohydrates then increases the viscosity of the juice. Okay, and then obviously if the viscosity increases, the flow rate becomes slow, so it affects the process and also the desirability of the actual beverage. So by using pectinase, the pectinase is used to clarify the juice, to get rid of the clouding, and also breaking down those insoluble carbohydrates and then decreasing the viscosity and having a free-flowing juice. Another thing that pectinase does is the pectin is usually found within the skins and within the fruit. So what happens is that it kind of is found in cells, and then cells that are sort of covering cells with juices inside. So what happens during juice extraction, some of the juice is still embedded within those pectin structure within the fruit. So by using pectinase, you degrade the pectin in such a way that you increase the juice yield. So all the juice that was also hidden or clogged or packed within there, everything becomes extracted. So you actually increase the juice yield, and increasing juice yield means that you have more money because there's more volume in terms of what can be sold. And then lastly, one of the most important thing is in terms of your pectinase, sometimes it's used so that it liquefies the entire fruit. So the entire fruit becomes liquefied, and then that is actually good in terms of maximum utilization of raw materials. So there aren't any wastes because the entire fruit is then used. And then we also have the cellulase, also in the fruit juices, but mostly in agriculture, whereby cellulase is also used for the removal of soybean coat. You know, the coat on the soybean, for soybean processing. So by using cellulose, it degrades cellulase, it degrades cellulose, and then the skin is loosened. You did that process in food technology one. I just forgot the name of the process. And then you lose those soybean coat via aspiration and rubbing. Okay, that's what I remember in food technology 1. And then other beverages, other important enzymes in other beverages will be the phenolases or polyphenol oxidase. Polyphenol oxidase, as we already mentioned, is an oxidation reduction or oxidoreductase, which is involved in oxidation reduction reactions, and it results in what we call enzymatic browning or browning of fruits, which can be detrimental in those fruits. If the apple turns brown, obviously nobody wants it, and it happens in fruits, vegetables like your lettuces, and then in seafood like your shrimp also browns. However, as much as it's detrimental in in fruits and juices and vegetables and seafood, it is beneficial. It's a beneficial fermentation process in the manufacture of tea, of cocoa beans for coffee, and then also coffee beans and cocoa bean for chocolate fermentation. So it results in the fermentation of your green cocoa beans, and then they are ready for roasting. So it becomes beneficial there. So as I already mentioned, it is used, or the endogenous polyphenol oxidase is relied on when your green rooibos tea is fermented to red rooibos tea. Okay, and then lastly, we've mentioned about these enzymes in these different processes, but what are the benefits there for? So there's just 2 types of benefits. We have what we call the processing benefits and environmental benefits of using these enzymes in the food industry. This is more like recapping what we've already mentioned. Number 1, we mentioned the processing benefits. It reduces the viscosity. I mean, that viscosity would have been due to your pectin and other polysaccharides that are found within the fruit during juice processing. It improves extraction, similar to the juice processing, whereby we mentioned that it increases the yield. And then it carries out bioconversions. Bioconversions, we already mentioned biological conversion of your catalase, which converts your hydrogen peroxide to safer water and oxygen. So in that way, we don't have the toxic hydroperoxide. We also mentioned lactose. Lactose has been broken down into glucose and galactose for those people that are lactose intolerant, using the enzyme lactase. It also enhances separation. And then we mentioned enhancing separation, as in we mentioned that in the meat industry, where you have some of the meat scraps that are adhering into bones, those are then used separated from there because of the degrading of the proteins. Or it creates or intensifies the flavors. We mentioned creating and intensifying the flavors in terms of the Maillard reaction. We mentioned that the fact that your amylases and your glucoamylase can break down your maltose into sugars, into simple sugars that act as reducing agents that can then be beneficial for the Maillard reaction. So it forms flavor and color. And also we mentioned that your lipases are used in the maturation of cheese, whereby it breaks down the triglycerides into free fatty acids that bring about the flavor in cheeses, especially extra matured cheddar. And then lastly, it removes harmful chemicals. We can also say lactose is a harmful chemical to people that cannot utilize it. But importantly, we had mentioned about acrylamide that can form if the amino acid asparagine is present and the protein reacting to form acrylamide. But if you use asparaginase, it breaks down the asparagine and it's no longer available to form Maillard reaction via acrylamide formation. And then we come to the environmental benefits. Environmental benefits, obviously, it has to do with pollution and everything else, or reduced chemical usage. So we don't need to use HCl anymore. The enzyme can be used. And when using HCl, we already mentioned that it's actually coupled with high temperatures. High temperatures means a high energy consumption, and also then there's also less greenhouse gas production, less corrosive waste, because remember HCl, after it has done whatever it does, it goes into the drain. When going to the drain, it goes into the oceans and dams, and then obviously we know what it does to the fishes and other small organisms that are living in there. And then we even say less corrosive waste, because some of this HCl can actually be corrosive to the equipment that is used in the industry, so it can start eroding the mixing vessels or the processing vessels. And also this then will reduce wear and tear, because then if those vessels that are used for processing are being corroded, it means you have to buy more and more, so that's wear and tear. And then also very important, because the people that are involved in food production are much safer now, because they don't have to inhale the fumes due to your hydrochloric acid or sodium hydroxide or any other chemicals for that matter. So thank you very much for listening. This will be the end of the recording.